I think we're just gonna go without it for
Yeah,
today.
I think that's fine.
Yeah,
That's cool.
yeah. Okay,
So can viewers online see me?
no, that's the
I
problem.
mean, see the presentation.
They can
sure.
see the presentation,
Okay.
yeah.
Yes.
Are
I'll
you
see
sure?
In the department.
We'll figure it out
I'll
for
recognize
next
my
time.
voice.
Yeah,
But you're sure that people can see the presentation
yeah,
now?
because I just checked it on the other one,
Okay.
so. I'll let you bring your presentation back up.
Sweet.
All right. Good afternoon, everyone. Sorry for the little delay. We had some troubles setting up, but we're good to start now. So I have the privilege to introduce you to our speaker for today, Dr. Fletcher Halliday. Dr. Fletcher Halliday is an assistant professor in the Disease Ecology and Diversity Lab at the Department of Botany and Plant Pathology at Oregon State University. He got his distinct honors bachelor's in molecular environmental biology from University of California, Berkeley, and his PhD in biology from University of North Carolina, Chapel Hill. His research focuses on the community ecology of infectious diseases, plant community ecology, and species invasions. Dr. Halliday is also a dedicated teacher and mentor, actively communicating his research to practitioners and the public via workshops, presentations, and social media. Today, Dr. Halliday will be sharing his work on disease risk under biodiversity loss and environmental change. Please join me in giving a warm welcome to Dr. Fletcher.
Thanks for the intro, and thanks for inviting me to come talk to you all. When I was invited to give this talk, I kind of freaked out a little bit. I gave a seminar a year ago at the EECB seminar. I gave my job talk the year before that, so I worry that my talk is getting a little stale. So I tried to create something new for this presentation, and what I want to do today is introduce you. I want to get rid of that bar at the top. I want to introduce you to my lab. Oh my goodness, you're kidding me. Well, you're gonna live with that, I guess. I want to introduce you to the people in my lab, and my lab group. We call ourselves the Disease Ecology and Diversity Lab. I'm going to talk about a little bit of the research that's been done by these three students in the group, and a little bit about what we're trying to get involved with here at Oregon State. So the students I'm going to talk about are Aram Kugler. She's in the top left. She just finished her master's at the University of Zurich, which is where I was before I was here. I'll talk about some work from Nicolai Zurbuchen. He's the one in the middle. He's a current PhD student in Zurich, although he'll be here for the month of November. So hopefully he gets a chance to interact with folks here. And then I'll talk about work from Bella Stuyvert, who's a current PhD student in botany and plant pathology here. Before I get started talking about my group, I thought I'd give you a little bit of context about why I'm interested in the work and what I do. I started my professional career not as a disease ecologist, but as a restoration ecologist, working for a consulting firm in the San Francisco Bay Area. And this is one of the restoration sites that I was working on. This is in Livermore, California, and we were trying to restore a degraded stream bed into a native grassland. You can see some flags here where we've planted grasses. We're planting willows over there. I was working on this project as I was thinking about graduate school. I had some background working with parasites, actually in invertebrate hosts, and I had some background in plants. This is another picture from that site. Does anybody know what grass this is? This is not a grass room. This is Sportium brachyanthirum. It's meadow barley. It's a native grass in California. It's also a native grass in Oregon. And there's something wrong with these plants. Can anybody spot anything weird in the picture? Well, they gotta be diseased, right? Because I'm going to talk to you about disease. So what I noticed, we're trying to restore a population of this plant in this field site. And what I noticed was that these inflorescences didn't have any seeds in them. They're full of fungal spores instead. These plants are infected with a smut fungus that has replaced their reproductive output with its own reproductive output. So I was pretty stunned when I saw this. My two interests are coming together. And I started searching to try and figure out what this was. I never got an answer, but what I stumbled on were some researchers who were studying pathogens in native grasslands in California. Two of them were here at Oregon State, Eric Seablum and Elizabeth Borer. And their collaborator, Charles Mitchell, was at University of North Carolina. And I ended up going there and doing my PhD with him and studying this kind of thing. It turns out that parasites are everywhere. They're under our feet. They're all around us. By some accounts, they make up 40% of all described species. They make up 75% of food chain links. And as a disease ecologist, I'm interested in trying to understand how the abundance and diversity of parasites is influenced by the environment that they live in. It turns out that these plant systems that I think are really cool are also really amazing model systems for studying disease ecology. So we have lots of data on plants at very large spatial and temporal scales. So we can look across large spatial scales to explore these questions. They're super tractable. We can build communities of hosts. We can put sentinel plants out into the field and they're exactly where we left them so that we can resurvey them with ease. And disease is readily quantified in plants. We can look at a leaf like this leaf and we know something about how infected it is and who's infecting it. It's really great model systems. Research in my group tries to understand how species interactions in the environment affect the diversity and spread of parasites. And our research is really carried out at two spatial scales. We're doing research at the scale of individual leaves, like that tall fescue leaf that's infected with Rhizoctonia solani. And in that research, we're really trying to understand what structures parasite communities within hosts. We also do research at the scale of whole host communities, like this grassland in southeastern Switzerland. What I want to talk about today then are three sort of big themes in our research group that's spanning those two levels. I'll talk to you about, can I do this over here? Yeah, okay. I'll talk to you about within host interactions across ecological gradients. This is a picture of a leaf of Dactylus glomerata orchard grass, and it's infected with a bunch of different fungal pathogens that you can all see with the naked eye right here. I'll talk to you about work that we're doing on global change, host trait variation, and disease risk, mostly in the Swiss Alps. So this is a cool infected plant in our Swiss alpine grasslands. And then I'll talk to you about some of the work that we're doing trying to integrate these ideas, thinking about how within host interactions might moderate or change or modify relationships between host biodiversity and disease. Okay, so I'm going to start with this. I'm interested in parasite interaction. So how do parasites interact with each other when they share the host? And one of the things we've discovered in plants is that these interactions can be antagonistic. Pathogens and parasites can compete with one another, but they can also be facilitative. Parasites and pathogens can increase the risk of infection. During my PhD, we developed a model system using tall fescue, this grass here, and 3 fungal pathogens or fungal parasites that commonly infect this out in the field. We use sentinel plants to track infections over time on plant leaves, and then we use that temporal data to infer how those parasites were interacting with one another. And as you can see, so I'm showing competition or antagonistic interactions with these red clubs and facilitation with the blue arrows. We see a whole suite of different kinds of interactions. The competition is not really surprising, right? These parasites, they're sharing the same host leaf. So they want the same physical area on that leaf. They want the same resources of that leaf. And so it's not surprising that they would compete with them. The facilitation is pretty interesting, though. And there's a lot less theory, or at least at the time, there's a lot less theory about facilitation within plants. And the other thing that's really interesting is that in any given relationship, you can have antagonism or facilitation occurring. And it often depends on which parasite shows up first on the leaf. So for example, we found that if this rust fungus gets on the leaf first, it reduces the risk of infection with polytoxicum, this other pathogen. But if polytoxicum gets in first, and then the rust infects, it actually makes these infections more severe. So really interesting stuff. As a postdoc in Annalisa Leina's lab at the University of Zurich, I was really lucky to kind of get a data set to play around with these ideas. My colleague Rachel Penskowski had done this really cool experiment trying to think about how the plant drives these interactions, the role that the plant plays. So what Rachel did was she took powdery mildew of different strains and she inoculated one leaf of a plant. This is Plantago lanceolata. She inoculated one leaf with the pathogen, and then she covered it with a mesh bag so the pathogen could not spread on the host. And then she had a mock inoculation where it wasn't inoculated, it was just covered with a mesh bag. And then she exposed all these hosts to really heavily infected hosts with a variety of different pathogen strains on them. What we found when we looked at the data was that those plants that had been primed, the plants that had been inoculated by some pathogen, were way more susceptible to subsequent infection by different strains. So something about that early infection was modifying the host immune system in a way that made it more, that facilitated subsequent infection by other parasite strains. So I don't just work with plants. I'm also interested in sort of generality of these different processes. And so over the past few years, I've been building a database of studies where people survey an individual host over time and measure parasite infection within those hosts. So this gives us temporal data on host individuals on the assembly and turnover of parasite communities within those. So I've been building this. We have lots of different systems. We have data from humans. This is my own field system from my PhD. We have African buffalo, which comes, some of that data comes from this university. Lots of stuff. We use that longitudinal data the same way I used it during my PhD to infer interactions. And so what we're looking at now, these are preliminary data from this survey. And what I've done is I've reported how the presence of one parasite in a host influences the risk that that host will be subsequently infected by another parasite. And then I've taken the data for all of those and analyzed it as a meta-analysis. So each of these points is a different interaction between two parasite species. When the points are above 0, that's indication that if that host is previously infected by one parasite, it's more likely to be infected by the other. So that's a facilitative interaction. And if it falls below 0, it's an antagonistic interaction. There's strong competition happening. And the first thing that strikes me when I look at this is that you have all the possibilities, right? All these different kinds of interactions are playing out across all these different systems. The other thing that's really exciting to me is that this is the grand mean, and it's well above 0. So it suggests that the most common relationship between parasites in this database, at least, is facilitated. Okay, so a big question then in my group that we're trying to grapple with is, why do we see so much facilitation occurring within hosts? And is there something special about the host environment that's driving this? Are these facilitative interactions disproportionately common in hosts compared to other kinds of environments? So last fall, Bella, a PhD student in my lab here at OSU, and I went to Leipzig to participate in a working group on priority effects, on interactions that involve the sequence and timing of events. And while we were at this working group meeting, we were talking with each other and with other people about this question. And that led us to, through those discussions, we ended up writing a little forum paper. These are really short review papers in Transition Ecology and Evolution, where we just kind of pose this question: are facilitative interactions disproportionately common in what we're calling symbiont communities, communities where some organism lives in or on another organism? And if so, why would we expect that to be the case? In the paper, we think about when we would expect to find facilitation. And the theory and the literature that we kind of lean on is this idea that facilitative interactions often happen when one species modifies the environment for another species. So through the process of niche modification. And that niche modification tends to be particularly facilitative in stressful environments. And sort of one of the characteristic examples of this are these cushion plants in the alpine tundra. So this is a very inhospitable environment. These early colonizing plants, these cushion plants, create a really favorable microclimate underneath them that allows all kinds of other taxa that otherwise wouldn't be able to be there to colonize and inhabit those areas. Okay, so kind of simple idea, two things. Niche modification has to happen for there to be facilitation, and that's more likely to happen if the environment's super stressful. Well, then we looked at symbiont community. And we realized that niche modification is almost required for symbionts to establish and grow in their hosts. So no wonder we see facilitation. And moreover, hosts are unbelievably stressful environments. They want nothing to do with you if you're a symbiont. And so we came up with sort of three classes of niche modification that we think are really common for symbionts, and three classes of stressors that we think most hosts have. And I'm going to walk you through really quickly, just an example of each of these, just because I think this is really fascinating. Okay, so one way that symbionts can modify their environment is through what we are referring to as physical modification. This is my favorite example of physical modification because I have little kids at home. So when little kids get a severe respiratory infection, their eustachian tubes swell and it causes their inner ear to fill with fluid, which provides habitat for bacteria, and that's an ear infection. So an ear infection is a really great example of physical modification. Infection by this respiratory virus completely changes the physiology of its host, and that leads to a new niche opening up for something that otherwise wouldn't have that. Another common form of modification that we identify in the paper is chemical modification. And maybe the most common version of that is something called syntrophy, where symbionts break down molecules in their hosts for food, and the byproducts of that breakdown become food for another symbiont. So in this case, can't remember the name of the bacteria. Bacteria breaks down peptidoglycans in the rhizosphere of a soybean plant, which facilitates later colonization by other bacteria. The third way that that symbionts commonly modify their hosts is through what we're referring to as biological modification. And sort of the characteristic example of this is immune modification. So here I'm showing a system that's been really well developed by folks here in African buffalo, where intestinal worms and mycobacteria can mutually facilitate one another by activating antagonistic immune signaling processes. So nematodes activate one signaling process, which downregulates the other and makes the host more susceptible to mycobacteria and vice versa. Okay, so like I said, hosts are also incredibly stressful environments, and I'm going to move away from the symbionts a little bit and think about why is it so hard living on a host? And one reason that hosts are really stressful is that hosts have barrier resistance. We have skin that protects us from getting things in and on us. We have mucous membranes that prevent things from sticking around when they get in us. Plants have thick, waxy cuticles, and symbionts have to overcome that barrier resistance in order to associate with their hosts. This is an example of ice-nucleating bacteria that form wounds on the surface of plants, which then allow other bacteria to get into those plants. Hosts have immune systems, and it's not just animals that have immune systems, plants have immune systems as well. So we think that facilitated interaction in Plantago was caused by downregulation of the host immune system, but it's also not limited to parasites. So this is a picture of an arbuscular mycorrhizal fungus. These are thought to be critical for many land plants for their ability to get nutrients. And there was a really cool study in 2014 that showed that for this really important symbiont to associate with its host, it has to at least temporarily downregulate host immunity. So that creates opportunities for other things to get in as well. And then last, hosts have a limited lifespan. They represent an ephemeral habitat, and symbionts have to complete their life cycles within the lifespan of their host. And it turns out that symbionts have a lot of different adaptations for coping with that. One group of those adaptations involves trying to make your host live longer. So this is an example of a rickettsia that modifies epithelial cells so that they don't undergo programmed cell death, so that it can complete its life cycle. And that can make the host more susceptible to bloodborne pathogens. Okay, I talked a lot about pathogens in these examples because I think about pathogens and parasites a lot. And I only gave you one example of each of these. So I want to sort of finish by talking about the fact that we scoured the literature and we found so many cases where these are taking place. So this is a supplemental table from that paper because foreign papers only allow you to have 15 citations. We snuck them all into the supplement. But what you can see is we're finding evidence of these mechanisms in plants, invertebrates, and invertebrate hosts across the mutualist parasite continuum, and they're showing up again and again and again. There do seem to be some gaps in our understanding. For example, this idea of barrier resistance, we weren't able to find any studies with invertebrate hosts that we could link to barrier resistance. The other thing that's striking is there are no studies that are testing all of these mechanisms, probably because we just made them up, right? But it means we don't know the relative importance of these mechanisms. And that's going to be really important if we want to start thinking about how interactions between parasites are going to change under climate change, what the consequences of this are for example, in the botany and plant pathology department for plant protection. And so what we're doing in our group right now is we're at the very beginning of trying to develop a new model system where we can explore these mechanisms. We're using this model host, orchard grass, Dactylus glomerata. I'm really excited to work with this grass because it shows extreme phenotypic plasticity, which means we should be able to get variation in those host-associated stressors. And it's globally distributed. It's native to Europe and North Africa, but it's present all over Corvallis, all over the US. We find it from the valleys to the Alps. That's what I'm showing on the left. This is a picture of it in coastal Oregon. This is a picture of it in this super arid environment in New Zealand. So it's all over the place and it's growing in lots of different environments. And so I'm really hopeful that we can use this to start to tease apart some of those host-associated stressors. And then Bella has been looking across a biogeographical and environmental gradient to try and look at some of the interactions that are happening in the microbes in these plants. So she's been culturing fungi across an elevation gradient in Eastern Oregon and in Switzerland, and she's hoping to test some of those mechanisms for how they facilitate one another with the cultures that she's been growing. Okay, that was a lot. Now I'm going to move on to global change, host trait variation, and disease risk. And this will be a little more brief. So for this part, I want to whisk you away to the Swiss Alps. This is where I've been working for the past 4 or 5 years before I came to Oregon. It's beautiful. But this is also a landscape that's defined by land use and by abiotic conditions. Biodiversity is being changed, it's being lost, and environmental conditions are shifting the distributions of species. This might look like a pristine environment, but this is actually a working landscape. All grasslands in Switzerland are working landscapes. Over here on the left side of the picture is a little herd of cattle. They're grazing this out. And there are studies, lots of studies on this mountain that are documenting shifts in species in response to climate change. We also know that these same factors, land use, biodiversity change, and the abiotic environment can dramatically influence disease risk. These are just 3 studies that have documented that, both empirically and through theory. And we're increasingly learning that host functional traits might serve as a nexus for connecting these things to one another. And the reason for that is that hosts seem to occur across a spectrum of form and function that kind of defines how they respond to their environment. This is a really popular spectrum of plant form and function that was published by Sanjay Diaz and colleagues. Those same traits that influence how host species perform in response to their environment are thought to impact how host species contribute to disease in the communities that they occupy. So this is something called the growth defense trade-off hypothesis, and it posits that host life history characteristics can be used to predict disease risk. On one end of the spectrum, you have hosts that are characterized by a slow pace of life. They're slow growing, they're very well defended, they have low leaf turnover, and they tend to contribute very little to disease in the communities that they occupy. On the other side of the spectrum, you have fast-paced hosts that are very poorly defended. Their tissues tend to be much more nutritious for parasites. And so they tend to contribute quite a bit to disease in the communities that they occupy. They're the more diseased hosts. And we often talk about them in disease ecology in terms of host competence. They're very competent hosts. They're very good at spreading disease. This idea has been used to explain disease in a lot of systems. Maybe the most popular place where we've seen this is in the dilution effect of biodiversity. This is an idea that was popularized by Felicia Keesing and Rick Ostfeld, who were studying Lyme disease in Eastern North American forests. And what they observed in their system was that hosts differ in what they refer to as host quality or host competence. On one end of the spectrum, you have high quality hosts that are contributing a lot to disease transmission. These are 2 high quality hosts in their system, the white-footed mouse and the white-tailed deer. And on the other side of the spectrum, you have low quality hosts that don't contribute very much. And what they found in their system was that as forests became fragmented and biodiversity was lost, the species that were sticking around were these high quality hosts, which meant that there was a lot more disease in fragmented forests in Eastern North America. The dilution effect hypothesis talks about the pattern that emerges from that. So when there's more diversity, there's less disease. This has been a really controversial idea in disease ecology for some reason. But when we did a meta-analysis on this idea, we found really, really consistent evidence in support of this idea that anthropogenic factors that drive biodiversity to be lost from ecosystems tend to favor those highly competent, high-quality hosts. So that's what I'm showing here. So when we looked at studies where a biodiversity gradient was associated with some anthropogenic driver, it was causing biodiversity to be lost from an ecosystem, we saw really strong increases in disease rates. When we looked across natural biodiversity gradients, where you don't necessarily expect one kind of compositional shift or another, we saw an idiosyncratic response. We saw responses that could go up to it. We've used this same idea to explain disease across temperature gradients. So this is from a study that we carried out in Switzerland along a mountain gradient. And what we found here was that as you go from cool temperatures at the top of our mountain to warm temperatures at the bottom of our mountain, the communities shift from being dominated by slow-paced species. So this is a physiological trait called specific leaf area, which we use as a proxy for host pace of life. So you move from slow-paced communities to faster-paced communities that are being dominated by species with a fast pace of life. And then we see that those same communities, the slower-paced communities, after accounting for variation in temperature, have much lower disease overall than the fast-paced communities. So what this is telling us then is that maybe we have a framework where we can predict disease in response to a global change driver by thinking about how that global change driver influences the distribution of host traits. So maybe perhaps host traits mediate this relationship between global change drivers and disease. If this is true, this is really cool because it gives us an opportunity to start predicting disease in the face of global change across a huge variety of different drivers, from forest fragmentation to climate change. So while I was in Switzerland, we got a grant to explore this. And the way we explored this was we tapped into this long-term research program in Switzerland. They call it ALEMA, and it's a long-term monitoring of biodiversity in farmland in Switzerland. And what they've done is they've put a grid of points all over the country in the main agricultural zones, and they resurvey those points they conduct biodiversity surveys every 5 years across these agricultural zones. I'm really fortunate because I like to work in grasslands, and grasslands are the most abundant agricultural type in Switzerland. Grasses are produced for forage and hay primarily. It's also a really cool system because they can quantify land use readily. So they can use NDVI, they can use remotely sensed imagery to assess how frequently these grasslands are being mown or grazed. And that mowing or grazing frequency is a really great measure of land use intensity. So that's what this is a map of. These red areas are areas that are being mown or grazed very, very frequently. They're very intensively managed. And these blue areas are what they call out there extensively managed. They're much less intensively managed grasslands. The other thing that is really striking when you look at something like this, if you know Switzerland, is that these areas really correspond to the environment as well. So this red area, this is the Swiss plateau. It's relatively flat and low-lying. And these blue areas, those are where the Swiss Alps are. They're steep, they can be quite rocky, they're high elevation and much cooler, they have shorter growing seasons. And so that the abiotic environment is actually driving land use intensity in this country. There was a really cool study done using the ALEMA data set by Eliana Meyer and colleagues, where they used structural equation modeling to quantitatively link these things to biodiversity. And they found a really consistent effect of abiotic conditions and land use intensity on farmland biodiversity. So we know that these things can impact biodiversity. So we went out into these meadows and we measured functional traits of plants. We measured specific leaf area, this proxy for pace of life, and we measured disease on a whole bunch of plants. And the first thing we saw was really good evidence for the first half of this model, that global change drivers can impact the distribution of host traits. So here I'm showing how dominated a community is, or sort of how fast-paced the community is on average, the community-weighted mean that hosts a specific leaf area. And we see that in cool climates, we have much slower paced hosts than in warm climates. Okay, so really strong distribution of this trait across a temperature gradient. We find evidence that that temperature gradient is really strongly linked to land use intensity. So here on the x-axis, I'm showing the annual temperature. And we can see that extensively managed grasslands tend to be much cooler than intensively managed grasslands. I mean, we could see that visually on the map, but it's nice to see some data to illustrate that. And it's good for us that there's some overlap here too. So they're quite different, but they also overlap in some important ways. And then when we look at the relationship between land use intensity and specific leaf area, this trait, you see a really strong relationship. So intensively managed grasslands are way faster paced. They're dominated by fast-paced species compared to extensively managed grasslands. So this all kind of fits with this conceptual model that we're building. And it's kind of intuitive, right? Like they're being grazed, they're being fertilized, sometimes they're being planted in these intensive managed grasslands. And you want your livestock to be eating yummy, palatable, fast-growing plants. So this isn't that surprising to me. Here's something that is surprising. We don't see the relationship between the traits and disease when we look at this, at least not across the whole data set. When we look at the relationship between host community, pace of life, and disease in extensively managed grasslands in these sort of more pristine habitats, we see the relationship that theory would predict. We see that faster paced communities have more disease. But when we look in this intensively managed grassland, we see it's either no relationship or a negative relationship, which we would not have expected based on theory alone. So that's pretty surprising. That was pretty surprising when I first saw it. This is looking at disease at the sort of scale of a whole community. We see the same relationship if we look at just one plant species, for example. So this is Festuca rubra, which is the most abundant grass in our data set. And we can see that if Festuca rubra is growing in a faster paced community versus a slower paced community that's extensively managed, we see this increase in disease. But if we look at it in intensively managed grasslands, we don't see that relationship anymore. So what's going on here? What we think is happening here is that this driver, this global change driver, in this case, land use intensity, it's not just creating this response, this mediation response, but it's also interacting with its own indirect effect. It's modifying its own indirect effect. And as I've thought about it more, I think it's kind of intuitive. The way that the land is managed out there involves complete removal of all of the biomass from the system. All of the plant and pathogen biomass is being removed. So it's not really surprising that that disrupts this ecological relationship. There are no pathogens left to infect anything because they have to recolonize those habitats from somewhere else. So Nicola has become really interested in trying to understand this disruption and how this might occur. And he did one really cool experiment looking at this. I don't have a lot of time left, so I'm not going to go into great detail on this. What Nicola did was he collected seeds from 3 different elevations on one mountain, and then he grew up those plants and transplanted them into different elevations. So he carried out a reciprocal transplant experiment on the mountain. And what that does is it means that a plant that's adapted to a cool environment is experiencing climate warming when it's planted in a warm environment, and a plant that's adapted to a warm environment experiences climate cooling when it's planted higher up on the mountain. And he sees this same sort of relationship. So what we're finding in this study is that warming increases plant pace of life. These plants become faster if they experience warmer environments. Now we're looking within a species too. So we're looking at a plastic response. But the traits don't predict disease in a system. So the pace of life of a plant just doesn't matter when we look at disease in these little, in this experimental context. Instead, what's driving disease is local adaptation to pathogen pressure. So at the top of our mountain, there's very little disease overall. At the bottom of the mountain, there's a lot of disease overall. And the plants that are adapted to that low disease environment, they're more susceptible to disease, regardless of where you put them. So this line of high elevation seed sources is higher than the other lines here. So local adaptation was driving disease risk in this, and the abiotic environment are driving disease risk in this experiment rather than this shift in functional traits. Okay, so when we take that together, it suggests that global change can define the context through which these biotic and abiotic factors drive disease risk. And this is really important because it means that that framework that I pitched probably isn't the best framework. It means that if we want to predict disease in the face of global change, we have to think about the particular global change drivers of interest and the biology of those systems. I guess that's not surprising. Okay, so here in Oregon, we've started a project trying to leverage the diverse uses of grasses around the state to think about the implications of this. And what we started this last spring and summer was a survey of these different grassland types here in Corvallis, which was really fun. We surveyed the lawn at the MU, we surveyed the rugby field, we surveyed the golf course across the river, and a bunch of pastures that are run by Animal and Rangeland Sciences. And we're hoping to use this to understand how these processes drive transmission of diseases across different land use types, so within and across land use. So what's the implication for all of this in terms of how diseases spread in the environment? Okay, 1 minute to talk about Aram's project. Aram has been doing this really cool project with this soil-borne pathogen called Rhizoctonia solani. It causes a disease called damping off. It kills seedlings. And so it means that we can plant seedlings, and this is on a poster outside my office, which is on East Cortlandt, so hopefully some of you can come take a look. So it kills the seedlings, which means we can plant a bunch of seedlings that are the same age, and then we can watch them die in like a chronosequence as the disease spreads through the single population. So this one, which we inoculated with Rhizoctonia, it didn't make it past the cotyledon stage. This one started to produce its first seed and then it died, or its first leaf and then it died. This one got a little more leaf produced before it died. And this one got big enough that it outlived the pathogen and survived the epidemic. What she's able to do from this then is quantify competence of a host species. How good is that host species at supporting this pathogen as it spreads through a population? And then she has a single value of competence for a bunch of species, and she can construct communities that differ in their competence and species richness. So she did that in our greenhouse in Switzerland. Each of these pots is a different mesoplasm. It's a different plant community. And then she layered over that access to this arbuscular mycorrhizal fungus. So this mutualist that plants have. So she's asking, how do host competence and species richness interact with the presence or absence of a mutualist to influence an epidemic that is carried out in each of these pots? And what she found here was really cool. She found that in the presence of arbuscular mycorrhizal fungi, host community competence, this is just the average competence of the species that she put in the community, is a really good predictor of disease. We would expect it to be a really good predictor. But when that mutualist is lost from the community, the relationship is disrupted, the relationship disappears. We know that biodiversity loss involves co-extinctions, and so this is another situation where we need to think really critically about what's happening under global change and how that might violate our expectations based on community ecology theory. So just one way that we're hoping to sort of take this to the next step in Oregon, the species that she used in that experiment, they're all native to Europe. The pathogen strain was a European pathogen strain, but all of those species are naturalized in Oregon. The pathogen, that pathogen can be found in Oregon. And I'm really excited to start doing some experiments looking at how local adaptation between hosts and their pathogens might be influencing both host competence and also the relationship between competencies. So that's something I'm excited to do next. All right, I'm just going to end with this slide. We have another working group in Germany where we're trying to expand this idea beyond just parasites and pathogens to think about all the things that consume plants. And we have our first meeting in the spring. And one of the things that came up during our meeting was that these things that we're calling biodiversity consumer relationships, how do the biodiversity of consumers impact plants? How does the biodiversity of plants impact consumers? They really sit at the heart of ecology and evolution. And I hope that means that by studying these, I'm placing this lab group in a situation where our work will be relevant and continue to be relevant to ecologists and evolutionary biologists for a long time. You know, if we want to make predictions about climate and environmental change, we probably need to know something about plant consumers and biodiversity. Biodiversity consumer relationships are instrumental for modern agricultural approaches, from biocontrol to using diverse mixtures of genotypes to prevent disease. They're important for understanding invasive species. They're important for understanding how species coexist and what maintains diversity in natural ecosystems. They're important for understanding the relationship between biodiversity and ecosystem functioning, for predicting and preventing the next pandemic. And so I'm really excited to have a lab group here where we can explore these biodiversity consumer relationships, hopefully in a way that's relevant to our colleagues here at Oregon State. All right, I'll end there. I want to acknowledge all the people who helped get this work done, especially the folks in the Disease Ecology and Diversity Lab and all of you. And I'm happy to take questions.
You talked about whenever you mentioned the hosts, the better hosts and those that remain, what was the reference that you did?
Sorry, say that again.
In the first section, in the first third of your talk, you mentioned the paper that was showing the hosts for symbionts or parasites that were better hosts.
So I think you're thinking about, so in the dilution effect literature, there's this idea that when forests are fragmented and biodiversity is lost, those really competent hosts are the ones that stick around. Is that what you're thinking?
Yes,
So you could look at Keesing and Osfeld, no, Keesing, Holt and Osfeld, 2006, Ecology Letters. That's a review paper. Here. Do I have? Yeah. So this is a review paper where they first really pitched this dilution effect hypothesis. And then the actual empirical work that underlies this happened in the 90s. But it's kind of fragmented, so this is a good citation to start thinking about.
So I was very disturbed that your final comment or one of your final comments about how all the parasites have also arrived with all these. And I really thought that through. So then if you go up to, quote, pristine environment in Oregon, can you find evidence that these introduced parasites then are invading native habitats?
So I think the questions about introduced parasites, those are outside the scope of the work that I've been involved with in the past. There are certainly very important introduced parasites and pathogens in plant communities throughout the US. The chestnut blight is the one that first comes to mind, right? Which completely reshaped Eastern North American forests after it was introduced in a New York botanical garden. Sudden oak death is another one, and that's touching, is that in Oregon? Yeah, okay, so there's another, that's an invasive pathogen. We did a study during my PhD where we looked at the microbiome, the fungal microbiome of grasses in California, both native and introduced grasses. We looked at 11 different species. I can't tell you much about who those fungi are, but I can tell you that we found fungi that could infect all 11 species. So we know that native and introduced grasses are able to share fungi, both fungi that are known to be able to cause disease and fungi that have never been associated with disease. We call those endophytic fungi. I don't know which of those fungi are native or introduced, though. I think the general thought is that when a host species is introduced to a new area, it leaves behind its parasites. And that release from enemies is thought to be really important for the success of invasive species. So when I'm thinking about like why we need to understand biodiversity consumer relationships to understand invasive species, it's more through the lens of enemy release, the loss of those enemies, than through like bringing in enemies that might spread to other hosts.
I feel like I need like 3 more hours to think about it anymore.
Yeah, it was a
You
lot of
just-
content.
It's very cool work. I'm just curious with your facilitation work, since that's, correct me if I'm wrong, but it seems like it's a meta-analysis kind of approach of existing literature. It just seems so complicated with, this kind of 6 different mechanisms, and I'm an ecophysiologist and think a lot about like the actual mechanisms underlying those things. How much do you worry that there is a publication bias in terms of what gets published more often, facilitation versus like not?
Yeah, so we opened that paper with a discussion of this idea that's becoming more popular in ecology, at least, that facilitation has really been underrepresented in it. That there was this great paper that was published by Anita Simha and colleagues, when was this, a couple of years ago, in American Naturalist, that argued that like human society and the way that we think about interactions has led us to focus on competition, like tragedy of the commons. So we study competition and we think that that's the way the world works, and we've imposed that on our research. And so there are people out there who are thinking about this and arguing that we actually are understudying facilitation. And I guess I would argue that like so the tree paper, it's just us thinking, it's not like, there's no data supporting any of that really. The meta-analysis that I showed though, that's completely unbiased, right? Most of those studies that I grabbed, they weren't looking at competition or facilitation among pathogens. They were tracking, like some of them were tracking the sociology of chimpanzees. And they just happened to be surveying those chimpanzees for parasites. And so I can use that data. So I'm hoping that that means that that data set's kind of free from bias in that way. Now, there might be bias in the way I analyze the data that could be leading us to more facilitation. And I'm a little bit concerned about that, which is why I haven't published this research yet. I'm not convinced that the analytical method is the best analytical method there. And so I want to think about that a little bit more before I publish it. Yeah. Yeah. There was one snail. It was a bunch of work from Russia and a freshwater snail. Limnaea stagnalis. So these are trematode parasites that infect this snail. As an undergrad, I worked on trematode parasites that infect marine snails, but I couldn't get access to usable data from that system. They have a hard time tracking, longitudinally tracking host individuals. So I can get a good one. I don't have any amphibian data, unfortunately. Amphibians are hard to do mark recapture surveys on. I recently got a data set on desert tortoises from Nevada, but I haven't had a chance to put it in here yet. So there will be a desert tortoise that's a vertebrate though. Yeah, is that?
On following on the facilitation, you could also think that it might be just like damage caused by the initial infection might be contributing to the likelihood of subsequent infection. So I wonder if you can control for like other kinds of just damage or, loss of fitness.
Yeah, I think damage is a form of niche modification, right? Like if I dig a hole in the ground, that changes the structure of the ground. It changes which resources are available in the ground. And so different plants are going to colonize that hole that I just dug versus an area that I didn't dig. So if a plant gets damaged or host gets damaged, that's an environmental niche. That host is an environmental niche and it's been changed now. And so I would argue that that's a perfectly valid form of facilitation. Now you could imagine that there are lots of forms of damage that aren't associated with previous infection by something else. And that could create a cool way to study that form, right? Like if we poke a hole in a leaf, is it more likely to be infected? I think the answer is yeah, which I think is why that study that looked at ice nucleation found that damage to the surface of leaves led to new bacteria invading its leaf.
I guess it's whether there's like a special effect of the prior bacteria.
Like, is it doing something special beyond just
Right,
damaging
yeah.
the host? See, to me, I think that just that damage is cool enough. Like to me, that's a form of facilitation. Like you don't have to be trying to facilitate something else to be doing it, right? Like you're just trying to get a home for yourself. And in doing so, you're creating a home for other things. I mean, it's like, yeah, it's like the same thing beavers do, right? Uh-huh.
You know why reduced biodiversity favors high polycyclosis?
Yeah, so the idea for a lot of these ideas are linked to life history strategies. So in really disturbed environments, the thought is, and I don't actually know how much support there is for this, but this is at least the theory, is that in really disturbed environments, hosts with a fast-paced life history strategy tend to be more favored. So you can think about like an urban environment in mice, or a fragmented forest, again, in mice. So those life history strategies are being favored, and those life history strategies seem to be linked for some reason to disease. Probably because those fast-paced hosts are thought to invest less in their immune systems, or like in the Lyme disease system, they groom themselves less, and which makes them better for ticks, and it's a tick-borne disease. Yeah, I think it's a really cool question. It's a really important question to ask, and I think that a lot of people assume that this is the case, and there isn't necessarily great data to support that yet, because actually measuring competence is really hard, and actually measuring life history trade-offs is really hard. And there are concerns that, you know, it might not be a fast, slow world. Like there might be other trade-off axes that people need to consider. And so I think it's a really cool, big open area to explore. Hey, this was so great. Thank you for all the awesome questions. Oh yeah, and I'm on Cordley 4th floor, right at the east-west split. So come chat with me if you're interested.